Patseq Explorer. Welcome to the Patseq facility of the Lens, a facility created to enable transparency and access to genetic sequences found in patents. If you would like to examine and compare biological patent holdings in the Lens with those in national and regional patent offices, view sequence disclosures, or download sequence data from chosen jurisdictions, please try Patseq Data and view its explanatory video. To search for matches to your favorite sequence in our database, you will find Patseq Finder, a useful tool. To search by a species or a keyword, or both, please try the text search. If you are interested in navigating patent-disclosed sequences that are mapped onto genomes and chromosomes, we offer two new tools, Patseq Explorer and Patseq Analyzer. At present, we have a Patseq Explorer for the human genome and another one for the mouse genome. And soon we hope to release new explorers for maize, rice, and soybean genomes. In the Explorer of the human genome, sequences were mapped against the reference genome with varying degrees of homology. The default view depicts map sequences with 95% similarity and 100% coverage. On each chromosome, aligned sequences are grouped in bins of 10 megabase each. The number on the x-axis reflects the maximum number of aligned sequences in a bin within that specific view. Mapped sequences are displayed based on document type, grants or applications, and whether the sequence is referenced in patent claims. For example, the Grants in Claims category, depicted in red, represents those sequences referenced in the claims of granted patents, whereas the Grants category includes both kinds of sequences, those referenced in the claims, coloured red, and those just disclosed, coloured yellow. You can also view sequences referenced in the claims of patent applications, or those that are either referenced or disclosed in applications. You can investigate one or all four categories. You can filter by various similarity and coverage thresholds, by jurisdiction, and many other attributes such as patent publication or filing year, sequence length, or by declared species. Or you can search by various patent attributes. For example, searching in claims for breast cancer keyword shows that there are 1,438 map sequences across the genome that are referenced in the claims of 952 granted patents, and which contain the keyword breast cancer in the claims section. Suppose you are interested in the sequences on chromosome 17. By hovering over that chromosome, you can view the total number of sequences mapped on that chromosome and their corresponding patent document counts. Click on chromosome 17. Once in the chromosome view, you can interrogate the data, grouped now in smaller size bins, 2 megabase bins instead of 10 megabase. By simply hovering over each bin, you can view its content in a tooltip. For example, in the bin aligned to position 40 to 42 megabases, there are 23 sequences referenced in the claims of 5 patent documents. You can link to the 5 documents in the Lens, build them into a new collection, or add them to an existing collection. Remember to register and sign in if you would like to keep your collection. To explore further the 23 sequences at the genomic region level, you can view them in Patseq Analyzer. This tool allows you to closely examine these sequences and compare patent claims that reference them. Please check the Patseq Analyzer video for more details. Another interesting feature in the Explorer for Human Genome is the integration of the OMIM database along each of the chromosomes. Looking at the OMIM data in the 40 to 42 megabase bin, you can see the linkage to breast cancer. You can then link to go to the OMIM database website and examine the associations between phenotypes and genes, and learn more about the progress of research in this area. You can also search for a disorder or a trait within the Search tab of Patseq Explorer. With all categories, grants in claims, grants, applications in claims, and applications turned on in the genome view, let us search breast cancer. With the help of a drop-down menu, we will also select breast ovarian cancer, familial 2 and 1, and press Enter. Results show that on chromosome 13 and 17, there are associations with such genes. Zooming on chromosome 13 over each coloured category, you can view the number of sequences, their corresponding patent documents, and explore their nature and function in further details using Patseq Analyzer. All results in Patseq Explorer are embeddable in your own blog. By simply clicking on the Embed icon, you can choose the size of iframe you wish to use to embed in your own space. We hope you find the Patseq Explorer tool useful. Its purpose is to provide a higher level of transparency about sequences disclosures in patents and more integrated sequence and patent searches at the genome level and across various chromosomal regions. For targeted analysis of patent sequences at gene level, please watch our Patseq Analyzer video. Please send feedback to support@Cambia.org.